Good afternoon, everyone. Uh, so today I will be talking about ALK detection using NGS and the comparison of NGS-based approach with IHC and FISH standards. So first of all, we all know ALK is a major driver oncogene, and fusion of ALK most significantly detected along with EML4 is one of the driver alteration, perhaps one of the earliest driver alteration in lung cancer, which has been very well targeted. So in this fusion, what exactly happens is there is a So there is a chromosome 2 location for EML4 going in the left side direction, and ALK is going on the right hand side direction. During the translocation event, this internal part is inverted, which results into fusion of EML4 ALK together. And because of this fusion, the outer domain of ALK is lost, and instead of that, EML4 comes into the picture, which results into cytoplasmic protein of the fused protein, and this is losing all of its control, which is driven by the ligand in a biological reaction, because of which it is constitutively active and it is oncogenic in nature. Now, to detect this fusion, there have been several approaches which are the current standard practice. One is by using fluorescent in situ hybridization, sorry, in which either fusion signal specific probes are designed for gene A and B, and if they both are coming together into a particular location, that indicates gene fusion. Or we use break apart probes, in which on a single gene we design two different colored probes, and if they are falling far apart compared to each other, then that indicates the presence of fusion. This technique requires specific designing of the probes at a particular location, and how that becomes a limitation and how that is different from NGS, that we'll discuss further in the slides. But we need to remember that these probes are specifically designed for the sequence available to us. Now, there is another technique called immunohistochemistry, IHC, wherein antibodies specific to ALK C-terminal has been designed, and equivocal presence of this unequivocal presence of this IHC positivity in the tissue is has been observed to be very strongly associated with ALK inhibitor response, and that is one of the standard technique being used as of now. So again, this is antibody specifically designed to ALK C-terminal, and it doesn't tell us what is the fusion partner, what are the variants involved in the fusion partner, and how that can help in therapeutic decision making. In NGS, what we do is we take the genomic DNA or RNA of a patient sample, and we chop them up into smaller fragments, as can be seen here in these orange lines. And the smaller fragments are sequenced by a next generation sequencing machine. And after the sequencing, we align all the data to the reference genome, which is a standard human genome sequence. After the alignment, we find out the point mutations, insertion deletions, large deletions, as well as gene fusions. So the logic to identify gene fusion particularly is to identify gene read pairs which are aligning to one gene at one part, and another part is aligning to second gene or second chromosome altogether. This kind of signal clearly indicates that in the patient sample, whether it is DNA based or RNA based, these two different distinct genomic locations are coming together, and that's the only reason why we are able to detect a single read in part mapping to either of the location. So by using this logic, we identify gene fusions using NGS dataset. Now here we are not designing any specific probe for this location or this location or any other fusion location for that matter. So this is totally unknown specimen. If you obtain, this is totally sensitive technique to pick out any gene fusion which is present in that particular location. So now, considering these three different technical scenario, why we need to discuss about it? And I mean, it could have been a much more simpler approach. Why exactly ALK fusion detection requires such kind of complicated technical knowledge, and why do we need to go through it? So here I'm showing you NGS data published in Journal of Pathology in 2017, which shows EML4 ALK fusion variants, as can be seen here, with each of them having a relatively shorter length of the fused protein. There are some long variants of EML4 ALK as well, in which a longer fragment of EML4 is fused with ALK, and they all again create different kind of variants. Apart from EML4, there are several other genes which are known to fuse with ALK, and they all are also found to be sensitive in their respective manner. So these are only the fusion variants shown by the pictorial diagram. If you look at the actual genomic break point in this EML4 ALK variant, you get to see that so many different locations of EML4 have been found to be fused with so many different locations of ALK. So each of the line indicates presence of one fusion event in a one particular patient sample. So even within a same fusion partner, the breaking point of EML4 and ALK has been distributed across several different exons, and this plurality, this heterogeneity creates a lot of complication in interpretation of results and therapeutic decision making. So these are the major points which limits the piece-based detection of this fusion event, because piece probes, if they are by chance falling in this any of the fusion location, then a piece will fail to detect signal. However, IHC being independent of fusion partner, it is not having the same limitations. So that's why in the clinical practice, we have observed more sensitivity by IHC. Furthermore, in these fused patients, when they are treated with ALK positive patients, when they are treated with ALK therapy, we have observed development of resistance, and majority of these resistance mechanisms have been again a secondary mutation in ALK gene itself. Apart from that, we have observed some resistance mechanisms in ALK amplification as well as activation of alternative bypass pathway, but the majority of them have been ALK mutation driven. So this is another aspect where NGS can be useful. A comprehensive NGS panel will be able to detect the exact breakpoint fusion partner of ALK, irrespective of any of the gene, as well as it will able to detect any presence of resistance mutation, which can be present at the de novo stage even without the treatment, or it can be acquired mutation which happens after we start therapy. So NGS will be the only technique which will be able to pick up all of these alterations. So to summarize the findings, IHC is highly sensitive and specific. It is current clinical standard, and it is no doubt highly reliable. However, it has limitations of detecting the fusion partner and resistance mutation. Piece is very good in sensitivity and specificity, and it could be useful for confirmation of equivocal findings from any other technique, and but it will have the same limitation of not able to find the exact partner of the fusion and resistance mutation. NGS is sensitive and specific, and it will take less tissue if we compare it with the sequential and serial molecular profiling of single gene based test. Then we are on an average for lung cancer, we are wasting about 20 to 30 different sections of the FFPE tissue. If we go right away to the NGS, then we are able to do the entire assay, the whole spectrum of therapeutic alteration, just by using 5 sections of the same tissue, and it is able to find out the resistance mutation in the single assay without doing any other technique. However, NGS is at present still expensive and not reachable for many of the patients. So that is something that we all need to work upon and find out more solutions to bring down the cost. And it will also have limitations with respect to expertise and availability of machine at various different locations, which I believe will be again not a problem if we are able to bring down the cost. So this is the suggested flow chart for IHC. We know for equal results, PIS and NGS can be useful. For PIS, if results are less than 15% positive, then again alternative approach by IHC or NGS can be useful to confirm the positivity. And if we go for NGS, when it is positive, it is positive. When it is negative, it is negative. But there is one parameter we need to pay attention to, and that is coverage. If coverage of NGS reaction is low, then a negative result wouldn't be reliable. As I showed earlier in the chart, that multiple risks aligning to a single location gives us high confidence of finding. So in a typical clinical setting, if coverage is less than 500x, then only NGS negative result should be doubtful, and we need to go for any alternative approach, or we can repeat the NGS assay itself with better coverage. So that's all about comparative techniques which can be useful for ALK fusion detection. I'll be happy to take any questions. Thank you.
Thank you for the excellent lecture, sir. I have one small doubt.
Yes.
So in ALK, IHC, we can use as a definitive diagnostic test and can start the treatment. But in other, like ROS, for the NTRK, IHC is used as only screening.
Yes.
This is because of the possible false positives that protein. So why in ROS and NTRK, IHC is still a screening, while in ALK, it is a definitive diagnostic test?
So that's a good question. It has two aspects to it. One is that ALK being a classical gene fusion and driver, we have several different TKI in practice already, and several different resistance mechanisms have been detected, for which we know that we need something more than IHC to pick up all of those variants, ALK fusion variants, as well as ALK resistance mutation. In NTRK and all, it is still immature field. It is upcoming, and right now several of the drugs are in practice. So I believe over the time we will also get to learn about the various kind of resistance mechanisms and fusion partner heterogeneity in NTRK, which will again raise the same question that we are facing today in ALK. Second thing is that some of the fusions surprisingly have good consistency in terms of their fusion partners, and we still don't know the exact mechanism why some fusions have much more heterogeneity compared to the others. So NTRK is one of the such gene which has relatively much lesser heterogeneity as of now, but it is too early to comment on that with high confidence.
Yes, sir. So my question is regarding the clinical applicability, sir, because NTRK, definitely it's very rare. But ALK and ROS, as far as ROS and ALK is concerned, there is more than 60% homology between the TK domains. And so same molecules are also used for the targeted therapy. So if as a clinician, I will not be much interested in knowing the partner. But if IHC, because it is easily done test, can be done in the periphery, easily available kits. So while for ALK, IHC, it is acceptable to start the treatment, but we know that in ROS, which is very much similar, I cannot start treatment even if it is IHC positive. I have to send for the molecular test. Is there any molecular basis for that, or is due to the retrospective validation of IHC versus FISH in the ALK, which is not done in ROS yet? Is there any such?
So specifically about ROS, the IHC is again better antibodies needs to be developed for more sensitive and reliable detection. For ALK, we have, as I said, ALK is one of the classical oncogene, and several different resistance mechanisms and several different lines of therapies have been already identified. The same thing is still going on for other oncogenes, including ROS and NTRK. So over the time, the same question will come over there, and over the time we will come to learn that NGS is perhaps the better option if we want to capture the whole spectrum of sensitivity, whole spectrum of gene fusion partners, as well as the spectrum of resistance mechanism altogether.
Thank you, sir.
Thank you all.